Live streaming is on.
We can see your slides, Takashi. And can we hear you or not? Can you try to speak, Takashi? So now we see your slides, but we cannot hear you. So what happened when you joined the platform?
Maybe try to go back to the platform and see if you can unmute. On the video window. Can we unmute people, maybe?
Here. Can you speak again? No, it's no. I cannot.
Yeah, but now he can share already. Now we just don't hear him.
We have to start.
Can you hear me?
Now we hear you.
Can you hear me now?
Yes.
Okay, so can I try to share the slide?
Yes, you can share the slides if it works.
Yep.
Yeah, his video is off.
And your video seems to be off.
How about?
That's perfect. We can hear you and we can see the slides, and I think it's sufficient for the presentation. Yep. And for the discussion, we can make your video on. So can you launch the presentation?
Yes. Unfortunately, I can only share the browser window. So now this image is from the Microsoft Outlook directory. So maybe I cannot. Can I do that? Is it okay?
This is perfect. Yes.
Okay.
So we will make a small introduction. So please stay online as you are. You are all set. This is fantastic. Thank you. Thank you, Zeb from Dryfta as well for all the help, and Nicole and Romy, Nina and Jay. That's perfect. So good morning again. So we will now start session block 4, also with the on-site participants on functional ecology and metabolic insights. So the aim of this session is to go deeper into microorganisms and their functionalities. So not just about dynamics in microorganisms, but what can they do, this organism, where they are doing their metabolic activities, and how can we rationalize these microbiomes from population to functions. And we will start with a keynote presentation from Takashi Narihiro from the National Institute of Advanced Industrial Science and Technology, called AIST, in Japan. And Takashi is a group leader in microbial ecology there. And after Takashi's presentation, we'll have again 5 presentations of 10 minutes, where we can ask a rapid question after each presentation, and we follow with a breakout session of 20 to 30 minutes, as usual as yesterday. So Takashi, we are actively listening to you, and floor is yours. Thank you.
Okay. So yeah, thank you very much for introducing me, and I'm very sorry to late my presentation. So yes, today I would like to talk about simplify and understand the wastewater treatment microbiome. So yes, so I think many people perform the analysis of microbial community structures and their metabolic functions of several various kind of wastewater treatment plant by using 16S and shotgun metagenomic and RNA sequencing. So these high throughput molecular approaches are very powerful and have a potential to uncover the ecology of complex wastewater treatment microbiome due to the in-depth data set of DNA and RNA sequences. So yes, so in recent years, several big fantastic wastewater microbiome projects are conducted around the world. And yes, I think everyone loves wastewater treatment microbiome. I hope so. Yes, first example is from the Professor Zhou's group, 16S ribosomal DNA sequences from 1,200 active research samples. It's very big data set. And the second example is also very nice, and a 6-year survey is too long, and 46 anaerobic digesters located at Denmark. And they performed a much amount of the 1,000 sequences for the 16S. And the third example is, I think, very important, that connecting the microbial community structure to their function. So they recover the more than 1,000 high-quality MAGs, metagenomic assembled genomes from the Danish full-scale wastewater treatment plants. So I think these big data set are core of the MIDAS. MIDAS is okay pronunciation, and the big data. So they will present in this afternoon maybe, but maybe I cannot attend because Japan is very late night time. So I will follow after that. So the last example is our study. So I fortunately can have an opportunity to join the big microbiome data analysis project in the Professor Wenzel Liu's group at the University of Illinois, founded by JGI. So in this project, we recovered more than 1,300 high-quality MAGs and analysis the microbial interaction based on the genomic and metatranscriptomic sequences. So after I back to Japan, so I and colleagues are trying to catch up with this big and nice project by promoting a national project entitled Development of Novel Technology for Bioprocess Optimization in Smart Bio Industry, which has been launched in 2018. So our project mainly focused on the industrial wastewater, but not municipal sewage wastewater. So in particular, we need to focus on the food-related industrial wastewater in this project. So I show you some fact about the situation related to the food-related waste and wastewater in Japan. So there are more than 3,300 food or beverage processing factories, and they use a vast amount of water use. And the cost of industrial wastewater treatment is estimated to be maybe 5 to 6 times higher than sewage treatment in Japan. And yes, unfortunately, so big and large amount of food-derived waste are discharged annually. And some of these organic waste are treated by methane fermentation processes, but the usage or secondary treatment of digestate is an issue in our country because we have only limited land area for agriculture. So these facts indicate the importance of cost-effective and stable treatment of industrial wastewater. So here I show you an illustration for background of our project. So our project is not only for the wastewater treatment, but such kind of food supply chain. It comprises of 6 major parts like primary production, processing, transportation, distribution, consumption, and the treatment of waste. So, but in our project, we only focused on the wastewater treatment, and there are some issues available. So the instability of the process and a high cost to treat the industrial wastewater, and the operation in less manpower due to the decreasing the human population in our country. So we are trying to accumulate the data of wastewater treatment microbiome and associated operation parameters. And we intended to apply such kind of data set for the application of the novel operation control technology for wastewater treatment process. Yes, so far we collected a total of about 900 sludge and wastewater samples from industrial and municipal wastewater treatment plant. And yes, so I would say one thing is that, so I get some permission from several private companies that operated industrial wastewater treatment plants to show this presentation. So I think you may not be satisfied with some part of my presentation, but yes, please forgive me and I will try to my best in the presentation. So major target process type are conventional activated sludge, CAS, and also nitrification, denitrification, NDN. We collected approximately 400 samples each from CAS and NDN. In addition, 100 sludge samples are collected from anaerobic process like AD and EGSB. So here I show the method for microbiome analysis for our dataset. So for 16-nesting amplicon sequencing, we use some basically under standard method, I think, like we base bead beating and phenol chloroform extraction for DNA extraction, and primer set is for V4 region. And we use a MySeq sequencing platform for sequencing. For data processing, we use QIIME2 and Silver database as basic. So after the data processing, we use R or PAST4 for the statistical analysis and Cytoscape for the data visualization. And sometimes we use a PyClust2 for the function prediction based on 16S dataset. For the shotgun metagenomic sequencing, so yes, limited number, we can do the sequencing for the limited number of the sample. So these samples are representative for the each processes, likely 5 to 10 samples per processes. We use a NovaSeq platform, and for data treatment, so MegaHit for the assembly, MetaBat for the binning, and multiple gene annotation platform like Proca, Drum, and Keg Ghost Koala, and Merops database for peptidase annotation. So after such kind of analysis, we may get huge or large data set of the community profile and mags and metabolic function related to the wastewater microbiome. But yes, in many cases, it's too much to understand the microbial basics in wastewater treatment, especially for me. So yeah, our question is what types of organisms or metabolites are important in the process, or how to control such complex microbial ecosystem for stable and cost-effective operations. Yes, but yes, actually, I have not yet solved these questions, but so I would like to share my situation here after my presentation. So this is a flow chart to simplify the wastewater microbiome dataset in our case. So first, we focused only CAS and NDN process that had more than 500 sludge samples. So at the beginning, we found more than 800 families of microorganisms. It is too huge. So we try to define the core microbiome that are detected in all processes and having more than 0.1% of the average abundance in entire microbial community profile. So I'm not sure this definition is feasible or not, but I think this definition is maybe comparable to the core taxa in the MIDAS database. I hope so. So we found 97 families from the dataset, and then we performed the statistical correlation analysis with some wastewater parameters such as TOC, TC, TN, and ammonia, and also etc. So as a result, we find 24 families that having a positive correlation with carbon parameters. And also we found 20 families having negative correlation with carbon, and also 19 families is positive with nitrogen, and also 16 families having a negative correlation with nitrogen. So yes, and within the positive correlation with carbon parameter, 24 families, so there are 12 families that are having positive with carbon, but no correlation with nitrogen parameters. So it is indicate that these families may strongly affected by the carbon concentration in the wastewater treatment process. So within the 12 families, so these two families are uncultured taxa. One is the Pasei bacteria of the Pateshi bacteria, so known as OD1 group, and another is Xenobia of the Elaminobacteriota, formerly known as WPS2. So we try to analyze the metabolic functions based on metagenome assembled genomes and information from previously published known literatures. So I will explain our result in each step. So this plot is usual principal coordinate analysis plot of all of samples collected in our project. So yes, microbial communities of CAS and NDN are loosely clustered based on the weighted influx algorithms, like showing the left part of the plot. So we think we could find some commonality in the microbiome dataset from the CAS and NDN. So we found 812 families that comprising the 1,800 genera from microbial community dataset of our CAS and NDN processes. So bottom bar chart is relatively weak and it's difficult to see, but the bottom bar chart shows upper bar chart shows the detection frequency of each family. So family name is located in the bottom of the bar chart, but maybe you cannot see. But for example, the maximum is the family Commonodaceae that are detected from all samples, all 525 samples. It's interesting for me. And the bottom bar chart shows the corresponding relative abundance of each core families. For example, the maximum is the Commonodaceae with 8.2% on average. And the following, the Saporospiraceae and Rhodocyclaceae with around 6% population. So these families comprise the 1,800 genera, so it may be too much to understand the microbial community data. So next we defined core microbiome that are families detected in all 7 processes having more than 0.1% of the averaged abundance. So we can find 97 families that comprising the 517 genera. So this number is also much higher, but it's better to understand the microbial community structure using such kind of core microbiome analysis. So our core microbiome is distributed in 19 phyla. The majority is alpha and gamma classes of the phylum Proteobacteria, and Bacteroidota, Chloroflexi, and so on. So next, this heat map indicates the average relative abundance of each family, 97 families, on each NDN or CAS processes. So according to the heat map, also the population density pattern is different between the processes like that. But those core microbiome may play a very important role in wastewater treatment, including carbon and nitrogen removal. So we performed the correlation analysis between the abundance of core families and carbon and nitrogen parameters. So we first estimated the statistical contribution of carbon and nitrogen parameters, such as total carbon, total organic carbon, and total nitrogen and ammonia on the core microbiome population by NMDS plot. Here, the core microbiome population are significantly affected by the carbon, like a TC or TOC, and the nitrogen, like a TN and ammonia concentration, with a relatively low p-value, 0.001. So as a trend, core microbiome population in process A have a positive correlation with carbon or nitrogen parameters, indicating much higher wastewater loading rate in these processes. Whereas process number C or D shows a negative correlation with C or N parameters, indicating lower wastewater loading rate compared with the process number A. So yeah, next figure is very complicated, but I would show you microbial network analysis image based on the Spearman rank correlation coefficient values. We use 525 samples, and the network threshold is p-value less than 0.001, and the coefficient is more than 0.2. And it's very difficult to see, but the positive correlation are shown in red color bar, and the negative correlation shown as a blue color bar. So yes, just I highlighted the positive correlation between the carbon parameter and core families. So parameter is around the bottom of the big circle, like total carbon, total organic carbon, and also inorganic carbon here. And as a result, we found a total of 24 families that have positive correlation with carbon concentration. So similarly, we can find 20 families having negative correlation with carbon parameters, and also 90 families with positive correlation with nitrogen parameters. And the last is the negative correlation with nitrogen is for 16 families. So in summary, in total of 65 families of the core families have significant correlations with C, carbon, and or nitrogen parameters. So there are several patterns for the correlation. For example, positive with both carbon and nitrogen, 3 families here, and positive with carbon but negative with nitrogen, maybe around 10 families here, and also only positive correlation with carbon parameters, like here. So as I think interesting for me, so correlation-based analysis correspond to our knowledge, basic knowledge about the wastewater treatment microbiome. For example, under low nitrogen condition, which means relatively good nitrogen removal. So such under the condition, the denitrifier and the nitrifier were observed. Another example under the high carbon concentration, which means a relatively bad carbon removal in the process. In such case, sludge bulking related organisms such as Tetrasphaera or Caldidinea were observed. And yes, anaerobic Firmicutes bacteria such as Clostridiaceae and Streptococcaceae also detected under the bad high carbon concentration environment, indicating that they may affect the carbon concentration, and they can grow under high carbon environment in the process. And then here, two families of the uncultured phyla were also high significant correlated with the carbon parameters like Pasei bacteria and Xenobia. So as you can see, so correlation-based analysis uncovers remarkable microbes for organic substrate decomposition and nitrogen removal in the wastewater treatment processes. So next question may be, what kind of metabolic function is important for the growth or control of these kind of microbes? So hereafter, we focused on the metabolic functions of two uncultured phyla. Yes, so here I show the relative abundance in 16S community profile left side, and the right side is the number of megs for the 97 core microbiome populations. So we can retrieve a total of more than 1,100 megs from shotgun sequencing database, a dataset of Cas and NDN processes. Among these, so more than 700 megs are from core microbiome, which is accounting for 78% of the total. So yes, we have 5 megs of the candidate Pasei bacteria of the phylum Pathobacteria. Using drum annotation platform, we can see the 268 genes, but this number is quite low compared to the usual bacterial genomic analysis. So as for the carbon metabolism, CAGI in the carbohydrate active enzymes, glycolysis seem to be measure metabolic function, but much less function associated with the nitrogen and amino acid biosynthesis are observed. Instead, interestingly, so 41 peptides related proteins are detected from 5 megs. So how does Pasei bacteria acquire amino acid from the outside? So yes, this nice figure is from a publication from the Professor Banfield's group of the groundwater metagenomic analysis. In groundwater environment, Pathobacteria microbes may acquire amino acid and also hydroxybutyrate by using peptides. So they found several kind of peptides families from the Pathobacteria genomes. So in our case, from our wastewater treatment Pasei bacteria, we found 5 peptides families that were also detected in the groundwater metagenomes, like this magenta color. So there are so many peptide families were found from Pasei bacteria megs. So maybe wastewater Pasei bacteria may use specific kind of peptides to grow under high carbon condition, and they may try to balance the carbon and nitrogen nutrition by the activation of such kind of peptides. Yes, and after my talk, maybe Ms. Jelly talk about the importance of peptides in NDAMO ecosystem. I think it's very interesting point. So another example is for the Xenobia of the phylum Anaerobic. We have 11 megs. And using drum annotation, we found more than 1,000 genes that is 4 times higher than Pasei bacteria. So as for carbon metabolism, common pathways like CAGI and TCA cycle, glycolysis are observed. And also major pathways for amino acid biosynthesis are also found. And in addition, many genes associated with transportation and peptides are also found. So another nice figure from the publication from the Professor Fugenholz and Ferrari's group, they analyze megs of the candidates Aminobacteriota phyla intensively. So our 11 megs are assigned into the candidates Xenobia group, and according to their publication, Xenobia-related megs were found in acidic and industrial waste-related environments such as tailing pond, palm oil mill effluent, and the dechlorination bioreactor. So here I show the reported metabolic function of Xenobia. They are basically obligate heterotroph using several kind of substrate. and have some specific feature for the acidophilic adaptation and the protection against the oxidative and neutralizative stresses. So in our case, we found a polysaccharide biosynthesis system from the Xenobia megs. First, here shows a WSP signal transduction system, which can sense some environmental changes, like surface growth or mechanical pressure or something like that. And then they sense the environmental changes and produce cyclic di-GMP as a signal molecules. And another system is a PEL-like exopolysaccharide biosynthesis system, which can accept the cyclic di-GMP to produce extracellular polysaccharide, like here. And we also found another extracellular polysaccharide biosynthesis system. So according to these results, the wastewater Xenobia may use exopolysaccharide biosynthesis system to form biofilm or floc under high carbon concentration. So they may try to keep their biomass or their metabolic activity in the process. This is the hypothesis. So after my talk, also the importance of EPS in a phototrophic granule ecosystem is talked by Mr. Lucas. So I think it's also a very interesting point to discuss. So this is the summary of my presentation. So at the beginning, I had more than 800 families, and after the definition of the core microbiome, we have approximately 100 families. And by using statistical analysis, we focused on the 12 families for the positive correlation with carbon parameters. And also with we have a special emphasis for two uncultured fire that may use peptides or exopolysaccharide biosynthesis systems in the under the high nitrogen, low nitrogen concentration condition or high carbon condition. So as for epilogue of my talk, so many challenges remain for application development. So at the beginning of my talk, such kind of data-driven control system could solve the waste problems on site of the wastewater treatment plant, like instability or operation cost and manpower limit. So I think more data set is needed to develop such kind of application. And now, in last year of our project, we now try to accumulate the time course microbiome and some physical-chemical data in a pilot scale. So yeah, photo is here. Membrane bioreactor treating digested in Nagaoka City, Japan, where the methane fermentation process is operated for the food waste treatment. Yes, so yes, at the end, I would like to thank for the MIWI organizers, everyone. And also I would say thanks for the consortium for the smart bioprocess collaboration, for the several private companies and local governments, Nagaoka City and Saga City, and also Nagaoka University of Technology. So, and I also thank for the co-workers from AISD, Dr. Kuroda and Sato, Aoyagi, and Hori. They are they have paid attention to analyze the big data set. And also, I also thank you for the my funding program, SIP, from the Japanese government. Yes, that's all of my talk. So thank you for your attention.
Thank you very much. Warmest acknowledgement to Takashi. Are you able to share your video now so we can also see you for the presentations, for the discussion? I really liked what you made, the link with the other lecture of the session. So we will see the presentations of Gilly and Lucas Trebour and the others. So it's very nice when we have a keynote who is making the link with other speakers. Really, it's a nice message also for other presenters as well. And what is interesting is that you show what we can do as capabilities with amplicon sequencing and metagenomics, and what are the limitations as well. There will be some follow-up questions afterward. And you see, it's not just about correlation. We need to go beyond the correlations. We need to squeeze the mags, if we can say like that, to look at the metabolism, the metabolic pathways. Then across the day and at the end of the day, we should be able to understand how can we test these functions. So you mentioned, I make a direct question and then I give the word to Per. I think you had the question. So is it about more data or more biochemical knowledge that we need?
So yeah, so the biochemical or chemical data is very important to understand the microbial function. It's very important. But I hear the yesterday's session, so there some data sharing problem is occurred in our area. So yeah, it's very big problem or big problem to solve. But I think in my case, it's very difficult to share such kind of chemical composition or such carbon or nitrogen parameters. So maybe you cannot see any values of the carbon or nitrogen concentration in my presentation. So, but in such kind of presentation, the company give me some permission to presentation here, and maybe in future, so we can hopefully we can share our microbiome data with some specific chemical or some physical chemical data set. Yeah, so yes, it's very nice. Yeah, opportunity to show our results. So thank you very much.
Indeed, we need to make the link between the DNA signatures to what is happening biochemically in the reactor, and in between there is the metabolic area of the microorganisms, the physiology, the metabolism, and we need to address that with additional functional analysis. Per, you have a question, so you can use the microphone. So Per Halke Nielsen from Aalborg.
Is home. Yes. Okay. Yes. Thank you for a nice talk. It's really a very impressive data set you have. Lots of mags, lots of data. So I was wondering, you showed some correlations between the presence of different families and then a lot of operational parameters and plant parameters and so on. So you made the correlation on the family level. Do you think that is the right level to make those correlations, or do you think you should go to either genus or species level?
Yes, so it's very nice view. Yeah, it's I try to simplify the understand the microbial community. So now in my presentation, I use a family level. So after that, I dig in the genus level or species level or something ASV level. So, but I think the in wastewater treatment ecosystem, so the family level is the first step to understand the microbial community or their function, because after the shotgun metagenomic sequencing, so we can get a lot of metagenomic assembled genomes, but it is very difficult to assign to the genus or species level. So, but we can get some nice information at the family level using the MAG, I think. So I think the family level is very nice to understand on the first step, but we may need to more deeper insight. So we need to go to the genus or species level. Yes.
Yeah, because sometimes you do have different functions within the same family. Quite often, for instance, if you talk about PAOs and GAOs, you may have both within the same family, and they may behave very differently. So I think it's a good idea to go to at least genus and sometimes species. One other question, if I may. Yes, you focused on those two uncultured phyla, the Pateski bacteria, and another one. So, and I think that's interesting, and they have very interesting physiology. But are they abundant in your plants? Are they important in those treatment plants?
Yes. So I can show some data. Sorry. Yes. So here is abundance data. So based on the 16S, so the for example, Pateski bacteria in total maybe 1 to 2% of the population, and the Xenobia population is maybe less than 1%. But I think the 1% of the population is, we can say the important population in the wastewater ecosystem. How about you?
That's very interesting. Yes, thank you.
Yep, thank you very much.
Thank you, Per. Are there some questions from the chat, Nina or Shay?
Yes, there's a question from Mr. Changzi Wang. Do you maybe want to ask it yourself?
Are you online, Changxi? Or maybe just
Yeah. The question, well, first, thanks for the great presentation. And one of the questions is, what are the quality criteria of your MAGs?
Criteria for the MAGs, yes. So our criteria is, I may not show you. Yes, our criteria is completeness is more than 80%, and contamination is less than 10% is our criteria.
Okay, I think that's clear. The second question, did you find Passae bacteria or Xenobia in your 16S data sets?
Yes.
Yes.
Yes. Left side is 16S population data on average.
Yeah. And are they part of the core microbiome in the wastewater?
Sorry?
If these are part of the core microbiome in wastewater.
Yes. Yeah, core microbiome, but the definition by us. Yes.
Okay, great. Do we have time for another question?
Yeah, a final one, and then we keep the others for the Q&A afterward.
All right. There's another question from Mr. Ramanujam. Great presentation. Impressed with the globe of core microbiomes you showed us. Do you see any similarities among the phyla identified in different food processing wastewaters?
Yes, I have not yet the detailed analysis, but I think they may. I think they are very similar organisms in the process, in the different process. I think so. Yeah, I will update my information soon. Sometimes. Sorry.
Thank you so much, Takashi. And if you can stay online the whole session, at the end we will have a breakout with all the delegates, and it will be important to have you on board as well. So please stay online and see you then. So I will let the words to Nina Rothans and Jie Deng for the sharing of the 10-minute presentations now.
Yes. So first, we have a pre-recorded presentation of Lucas Trebur from Netherlands Institute of Ecology. And he's going to tell us a little bit more about functional analysis and community structure of photogranules.
Hello, and welcome everybody. My name is Lucas Trebur. I'm a PhD candidate at the Netherlands Institute of Ecology under the supervision of Tanja Fernandes and Marcel Janssen. Today I'm going to talk about a collaboration we did with Olivier Bourseau and Dirk de Beer from Max Planck Institute in Bremen and Thomas Neu from the Environmental Center in Magdeburg. The talk is entitled Deconstructing Photogranules, Microscale Functional Analysis and Community Structure of Phototrophic Microbial Aggregates from a Wastewater Treatment Reactor. First, what are photogranules? Photogranules are phototrophic microbial aggregates that we generated in a lab-scale setting, starting from a diverse inoculum of free suspended cells. Photogranulation was achieved by operating lab-scale reactors under sequencing batch mode, with 4 distinct phases of reaction, settling, draining, and filling. And by doing this, the microbial community aggregated first to flux, as seen here in this video. And by repeating this type of operation, the microbial community assembled finally 2 photogranules. Photogranules have excellent settling properties and appear as dark green rubbery spheroids, as seen here in the microscope. We investigate them for their potential for biological wastewater treatment and carbon, nitrogen, and phosphorus recovery. Photogranules are light-driven ecosystems and can use both light and carbon to generate metabolic energy. This leads to higher biomass productivity compared to conventional activated sludge or aerogranular sludge systems. And another important aspect is the potential for in situ oxygenation. Phototrophs produce oxygen that can be used by heterotrophs and in turn sequester carbon dioxide to reduce greenhouse gas emissions. For the study I'm presenting here today, we took photogranules of a lab-scale system that was operated on this specific conditions. Due to time constraints, I won't go into detail. But photogranules usually have a diameter of 0.4 to 5 millimeters. And we set out to investigate the microbial community, the metabolic functions, and how are photogranules structured on a microscale. We did this by selecting photogranules in the range of 2 to 4 millimeters, which were representative for the system and easy to work with. The microbial community was assessed with 16S, 18S amplicon sequencing. The 3D structure was determined by confocal laser scanning microscopy. Microsensors were used to investigate light, oxygen, and nitrate. Additionally, photopigments were extracted, and 14C and 15N tracer studies were carried out to investigate the carbon and nitrogen cycles. The microbial community, both for 16S and 18S, was characterized by a high abundance of phototrophic organisms. The 16S dataset was predominated by filamentous cyanobacteria, while 18S dataset was dominated by eukaryotic algae. Next to these, there were also nitrifiers and denitrifiers present. The physical structure of the photogranule was assessed using microscopy. We took photogranules, cut them in half, and observed the structure under white light, but also under laser microscopy. The CLSM image shown here has 3 distinct features. So in green, you see nucleic acids, basically all organisms. In blue and pink, you see the photopigments of cyanobacteria and eukaryotic algae. And in red, glycoconjugates, which represent extracellular polymeric substances, or EPS. Here, the same image is shown, but only in the channel for the photopigments. And this really shows and highlights also the arrangement of the filamentous cyanobacteria and how important they also are for the structure of the photogranule. Here in the next image, you only see the nucleic acids, which indicate kind of like a higher density of microorganisms at the surface. In the next slide, I would like to show you the same CLSM image, but magnified. When isolating the individual channels, again, you see the importance of the filamentous cyanobacteria on the photogranule structure. And here you see like this higher density of organisms closer to the surface. Finally, here are only glycoconjugates represented. These you couldn't really see like in the images before. Glycoconjugates are indications for EPS excretions by microbes, which act like a glue to hold microbes together. So further, we were interested in the light availability and how deep actually light can penetrate. As you saw before, cyanobacteria were throughout the whole photogranule, so we want to know where can they actually perform photosynthesis. So the light sensor showed that light was rapidly absorbed in the visible light spectrum, and after 0.5 millimeter, more than 90% of the light was already attenuated. This was done by an array of photopigments with chlorophyll A as the most prominent. Further into the photogranule, only light with longer wavelengths could penetrate. To get an even better insight, we performed 14C incubations to see the inorganic carbon fixed. By that, we could visualize photosynthesis, and we saw that most of the activity indeed was constrained to the outer 500 micrometers, which is in line with the previously shown light availability. We also did investigate oxygen profiles under light conditions with 3 different treatments. So measurements in tap water primarily showed photosynthesis with a maximum oxygen production around 500 micrometers. When using medium without acetate but ammonium phosphate, the oxygen consumption of nitrification could be observed. And when using the full medium, so with acetate COD, the oxygen consumption by heterotrophy exceeded the oxygen production by photosynthesis, which makes photosynthesis the rate-limiting process. So basically here, seeing an oxygen. The same technique of 14C incubation was used to, as for photosynthesis, was used to determine the nitrification activity. Similar to photosynthesis, also nitrification was mostly restricted to the outer 500 micrometers of the photogranule. The 15N tracer study also revealed nitrification and denitrification rates at different conditions. The highest nitrification activity was achieved in the absence of acetate as COD and with light, while the highest denitrification activity, overall 3 times higher than nitrification, was shown in the dark with acetate and the absence of oxygen. Interestingly, denitrification was also occurring at low rates in the absence of any external carbon source. This might be due to the use of internal carbon, stored carbon, for example, as polyhydroxyalkanoates, or recycled carbon from decomposition processes happening in the interior of the photogranule. Now, to conclude our findings, the structure of the photogranules was shown to be governed by a complex network of motile filamentous cyanobacteria and EPS excretion, of which both have an important structural role in the makeup of the photogranule. Additionally, a dense crust of microorganisms was found close to the surface. Further, we gained valuable insight into the microbial activities within the photogranule and how they are internally coupled. Photosynthesis was predominantly in the outer 500 micrometers, and in the presence of COD, the oxygen consumption by heterotrophs exceeded the oxygen production via photosynthesis. Nitrification, denitrification was also predominantly in the outer 500 micrometers, while denitrification also occurred in the absence of external carbon. Lastly, the question remained actually what is happening in the core of the photogranule, because only little activity was detected, and possibly there are decomposition processes occurring that potentially lead to internal nutrient cycling. With that, I want to thank everybody for listening and want to acknowledge all the co-authors, collaborators, and support staff, and of course, the funding agency to fund this research. I'm very happy to answer your questions.
Well, this is a remarkable presentation, and thank you so much, Lucas. Also, the quality of the images that you got together with Thomas and Ute. It's a very nice illustration of what can happen in these granules. I have a direct question about your metabolic methods. So what did you use as 13C and 15N labeling? So to detect it was nanosim. So what was the technique used for detection? Actually, I didn't ask if Lucas is online. Lucas, are you online? Yes. You should unmute your microphone. We cannot hear you. I don't know what. Maybe, yeah. So maybe you can type the question, the answer in the chat. But in short, what was the analytical measurement technique to detect 13C and 15N?
That this one work?
Yes.
Hi.
Yeah.
And I think he said, and also in the abstract I read 14C. So I was also wondering why was he using 14C and not 13C?
Okay. So it was radio labeled. So what was okay? So it was MAR with MAR microtoradiography that you did. Okay, so it was microtoradiography. Yeah. Next questions. I think they are from Maxime, for instance.
There's another one first from Mr. Ramanujam. Great talk. Can you share more details on the purpose of the 15N tracer study? This will be a longer answer, but maybe difficult to do it now via chat.
Yeah. Maybe what would be good is that for the roundtable afterward, you try to log out and log in again of the platform, and then on the top left corner of your web page, you have to validate the sound normally. So you have to give permission from your computer. Suggest that we move to the next speaker, but we keep the questions afterward for the roundtable. Great talk, Lucas. Thank you so much.
Yeah, so next we have another pre-recorded presentation from Ji Lee from the University of Queensland. And the presentation will be about microbial community interactions in biofilm systems coupling NMOX and NDAMO processes.
Good morning, afternoon, and evening, everyone. Thank you for joining my presentation today. My name is Jet Li, currently a PhD candidate at the University of Queensland. Today, I will introduce you our work about the metabolic network interactions of microorganisms in biofilm system, coupling Anaerobic and Endomo process. Recent years, there has been some updates of the conventional nitrogen cycle. Among them, Anaerobic and nitrate-dependent anaerobic methane oxidation are two significant changes. Anaerobic-based technologies have been widely applied for nitrogen removal. However, no matter for mainstream or sidestream, there are still a few concerns for Anaerobic process. For example, strict requirements for the influent nitrite over ammonia ratio, and the unsatisfied effluent total nitrogen concentration due to the nitrate production. How can we overcome these two barriers? Recently, because of the discovery of Endomo process, which is able to do denitrification using methane as carbon source, a new hypothesis has been proposed. Is that possible to use methane to enhance the nitrogen removal of the Anaerobic process? A novel concept coupling Anaerobic and Endomo process has been proposed thereby. In this combination, the nitrite could be removed by both Anaerobic and Endomo reactions, giving the system with the overcapacity to remove the extra nitrite. Therefore, the requirement for influent ratio will be relaxed. On the other hand, the nitrate produced from Anaerobic process could be reduced by archaea, so the nitrate accumulation problem can be solved and a good effluent quality can be expected. This is a beautiful idea, but how can we achieve it? To achieve this goal, two identity MBFR with hollow fiber inside for gas delivery were set up, fitting with mimic mainstream and sidestream wastewater with total nitrogen of 1,000 milligram and 50 milligram nitrogen per liter, respectively. Luckily, Anaerobic and Endomo co-capture was successfully enriched in biofilms in our laboratory. From the long-term performance of both reactors, we can see that extremely low total nitrogen concentration was achieved in both systems. So, who are responsible for such merit and how this happened? To answer this question, biofilm samples from both reactors were collected for metaomics study, including 16S RNA gene amplicon, metagenomic, and metatranscriptomic sequencing. Firstly, amplicon sequencing results confirmed the existence of Endomo and Anaerobic microorganisms. Meanwhile, there are some other functional and non-flanking partners existing in both systems, triggered us to think about who are these flanking partners and how they interact with others. The existence of Endomo and Anaerobic microorganisms were also confirmed by metagenomic data, which is the second and third column of the upper figure, where the y-axis represents different phylum and the x-axis represents relative abundance. The lower figure shows the dominating genomes recovered from metagenomics data with expression over 1%, which accounted for more than 83% of expression of both community, indicating majority of metatranscriptome were from the active genomes. It is clearly shown that different feedings shaped a different microbial community, no matter in DNA or in RNA level. For example, in MBFR1 with high loading, archaea dominated over Anaerobic over bacteria, while in the low loading reactor, an opposite order was observed. Interestingly, partner existed and also active in each system, triggers us the question, who they are and what are their doings in each system. In order to answer this question, nitrogen cycling and methane oxidation related genes were investigated as shown in this heat map. The color intensity represents the expression level of genes. The more red, the higher the expression comparing to the median expression of this genome. In the high loading reactor, the partner BAC2 is likely doing DNA with highly expressed NAR and NRF genes. In the low-loading reactor, since VR1, CHO3, and PRO7 are doing partial denitrification in the system. So partners were contributing to the nitrogen turnover in both systems. However, this triggers us the question, what is the carbon source or electron donor for these partners to do DNA or partial denaturfication, since methane is the only carbon source supplied into both systems? From the pathway profile, like aforementioned, Anaerobic microorganisms are capable of utilizing methane as electron donor. PiO7, one of the partner in low-strength reactor, is also capable of oxidizing methane with highly expressed PMO genes. Animals bacteria highly expressed the Wood-Longo pathway for carbon fixation. But what about others? The breakdown of EPS produced by animals bacteria has been proposed to be a major organic carbon source supporting heterotrophic growth in animals granulars. How about in biofilm systems? Expression of selected peptides were provided. Peptides possibly involved in EPS matrix protein degradation were showed. In high-strength reactor, BAC2 expressed quite a few outer membrane and extracellular peptides. In low-strength reactor, partners expressed more outer membrane and periplasmic serum peptides. Amino acid and peptide transporters were predicted across active genomes. Consistent with the peptides results, transporters of peptides and amino acids were more actively expressed in partners. This would allow the partners to use amino acids as carbon and energy source. The biosynthesis and the degradation of amino acids across each genome were investigated. We can see from the results that Anaerobic and Anaerobic microorganisms generated nearly all types of amino acids in both systems. Partners missed the biosynthesis modules for a few amino acids or expressed a higher level of degradation than the biosynthesis, suggesting extra external amino acids are needed for partners. Based on this analysis, the metabolic interactions for both novel engineered ecosystems were proposed. Here, we used high-loading reactor as an example. The upper left part represents Anaerobic archaea, the upper right parts represent animals bacteria, the lower parts represent the partner genome. Three major interactions among these microorganisms were highlighted, including cross-feeding of nitrogen, carbon dioxide, and organic intermediates such as amino acid, acetate, and vitamins. That's all for my talk today. Thanks for listening. Thanks for my supervisor team, Associate Professor Jianhua Guo and Professor Jin Tyson. Thanks for the financial support from the University of Queensland and all the amazing lab mates there. Thanks for organizing this conference. Let us have such an opportunity to know each other and the work everyone is doing. Thank you. Thank you all for listening. Welcome suggestions, comments, and collaborations. Thank you.
You have a lot of applause, Gilly. Thank you very much for your presentation. Are you online, G?
Yes, I'm.
Thanks. Hi, good evening. Are there some questions to G? Maybe some specialists of the nitrogen cycle want to ask a question inside the room? No? So I have a question about the metabolic maps that you showed. How can you construct them? So basically, many PhD candidates and senior researchers as us may think, okay, you have a metagenome, you get your bin genome, so you can get a MAG. How can you move from a MAG to these nice metabolic maps that you showed with this interaction between the two organisms? How can you construct that? Can you give some hints to other interested persons?
Yes. After we recover the genomes from the metagenomic data, we identify the active genomes by the metatranscriptome data, because we recovered actually plenty of genomes, like more than 50, but with the metatranscriptome data, we decreased these genomes, I think, to like 10 in total, because other genomes are not that active, and the genes were predicted. And for the interaction, for the pathways, I basically used the relationship from the CAG database, because this database will tell you the genes that's in the pathway. For example, the methanexation, which genes they used. Yes, and for the last part, the interaction part, right? For the interaction part, yes, for the carbon interaction, because methane is the only carbon that's supplied into the system. So, and for the partners, definitely they obviously they do not have the ability to oxidize methane. So tricks to think about the organic carbon. They are utilizing organic carbon from others. So that's why we analyzed the amino acids. Yes. Does that answer your question?
Yeah, very well. And then it means that we need to test if these reactions really happen in the organisms. And we will see across the with other lectures or other presentations how we can move forward with this functionality detection in microorganisms. Thank you so much, Chili. Stay online for the breakout session afterward, and we move to the next speaker.
Yes. So next we have Nadje de Jonge from Aalborg University, also a pre-recorded presentation about the differential Wood-Lyondel pathway utilization during in situ hydrogen biomethanation.
Hi, and thank you for the opportunity to speak about our recent research at the IWA Microbial Ecology and Water Engineering Conference 2021. Anaerobic digestion is an important pillar in our transition towards sustainable energy generation. An attractive technology within biogas production from anaerobic digestion is upgrading the biogas with hydrogen. The increased methane content in the upgraded gas allows for better integration into the natural gas grid, easier conversion into sustainable fuels, and has the potential for surplus electricity storage. However, the effects of hydrogen addition on an anaerobic digester and the biochemical pathway utilization by the microbial community that governs the anaerobic digestion process are currently poorly understood. Anaerobic digestion is governed by a complex community of microorganisms that through a cascade of biochemical conversions produce acetate, carbon dioxide, and hydrogen, which can then be converted into methane. Hydrogen is an important player throughout the entire anaerobic digestion process, but one pathway is affected more than most. The Wood-Lyondel pathway allows for bidirectional conversion of acetate into carbon dioxide and hydrogen, and vice versa, and hydrogen partial pressure directly affects this pathway. It is therefore hypothesized that the Wood-Lyondel pathway plays a central role in biomethanation with hydrogen. However, the effects of hydrogen addition to an anaerobic digester are currently not well understood. So we aim to investigate which microbial populations are active during anaerobic digestion and biomethanation with hydrogen conditions. To investigate this, we used metagenome-resolved protein-stable isotope probing, or ProteinSit for short. Laboratory-scale reactors were inoculated using sludge from a full-scale anaerobic digester processing manures and agricultural wastes, and allowed to operate under stable conditions until anaerobic digestion and biomethanation conditions were achieved by adding hydrogen to one of the two laboratory-scale reactor types. Once stable conditions had been achieved, batch reactors were set up, and stable isotope labeled bicarbonate was added as a proxy for carbon dioxide in order to trace newly generated proteins by incorporating stable isotope labeled carbon. The anaerobic digestion and biomethanation batch reactors were allowed to incubate for 48 hours, after which samples were collected for a metaproteomic analysis to identify which proteins were generated with stable isotope labeled carbon incorporation, and a metagenome was generated to investigate which microbes are present in the systems. Combining these data allows us to achieve sensitive detection of isotopic incorporation into proteins, while at the same time also achieving high-resolution microbial identification, allowing us to see which pathways were active under which conditions. The proteomics results showed that a much larger number of proteins had become isotopically labeled in the batch reactors receiving hydrogen compared to those operating under regular anaerobic digestion conditions. In total, more than 90 proteins were found to have incorporated 13C labeled carbon. These proteins could be assigned to diverse metabolic functions and were found to stem from diverse microbial populations, signified in the plots on the left by different colors representing different microbial phyla. Together, these results suggest a general shift in activity across the microbiome in the reactors operating under biomethanation conditions. A metagenome was generated based on the full-scale anaerobic digester used to inoculate the experiments in order to investigate which microbial populations were present in the reactors. In this graph, the size of each point designates the length of the DNA fragments present in the metagenome. The individual clustering of points indicates individual bacterial genomes that could be extracted from the metagenome, and their color designates their taxonomic assignment. When this data is combined with the proteomics results, here shown as purple dots overlaid on the metagenome to show which protein identifications could be linked to DNA fragments, it is possible to make a direct link between protein identification and microbial identity. Based on this information, we were able to extract 10 high-quality microbial genomes, of which 9 were shown to contain key genes of the Wood-Lyondel pathway after metabolic analysis. The Wood-Lyondel pathway was especially strongly labeled, and this made it possible to identify which parts of the pathway showed increased synthesis of new proteins and thereby activity. The batch reactors operating under biomethanation conditions were labeled on both ends of the Wood-Lyondel pathway. In the methyl branch, where carbon dioxide and hydrogen begin the conversion towards acetate, and near the end of the pathway where acetate is produced. Biomethanation conditions make it unfavorable for acetate to be converted towards hydrogen, and this therefore suggests that acetate was produced and potentially an increase in acetoclastic methanogenesis was observed. In the anaerobic digesters, only a few labeled peptides were found in peripheral pathways to the Wood-Lyondel pathway. This suggests that the 13C labeled bicarbonate was instead used for anaplerotic generation of complex compounds such as amino acids. In summary, metagenome resolved protein SIT was able to provide detailed insight into the identity and activities of anaerobic digester microbiomes operated under different operational scenarios. To our knowledge, this is the first study where it was possible to provide both a sensitive measurement of activity in a single pathway combined with a high-resolution microbial identification. Our study showed that biomethanation with hydrogen stimulated the activity of the Wood-Lyondel pathway and suggested increased acetoclastic methanogenesis. These results help us understand the microbial activities in anaerobic digesters during different operational scenarios, which in the long term will improve our management of not only the biogas facilities, but also the management of our microbial communities and our abilities to implement new technologies in full scale. Thank you very much for your attention. I'll be happy to take any questions you may have.
Congratulations, Nadi, for your very, very interesting insight, metabolic insight into these microbial communities. Are you online, Nadi?
I hopefully should be.
Yes, we can see you. Hi, how are you? It's nice to
Hello.
meet you again. So, are there some questions for Nadi right on the spot? Are there some questions in the room for Nadi? No? So I can make a question. So I didn't understand your conclusion on acetoclastic methanogens. Because if I understood, with the H2 biomethanation, you are looking at the hydrogenotrophic methanogens. So how did you come to this conclusion on acetoclastic ones?
I realized after the recording was played that there was one observation I actually forgot to mention. I mentioned that we found 10 labeled mags of high quality, and one of these was actually one of the acetoclastic methanogens, methanosacina, and we also observed some labeling in Methanotrix or Methanosaeta at the same time, which leads us to believe that combined with the production of acetate, it is likely that the microbial community shifted its activity from hydrogenotrophic methanogenesis towards the acetoclastic pathway, if that makes sense.
Okay, thank you. There is a question from Maxim in the chat.
There's another question before that from Mr. Sohail Neshat. Great presentation. How can the labeled proteins be traced back to individual mags?
So the primary reason we generate the metagenome is to identify which microbes are there, but also to also do the metabolic prediction on them, to generate a database to assist us in identifying the proteins. This way we can link the we can link the individual mags or scaffolds in our metagenome directly to the proteomics results.
Yeah. And finally, there's a question from Maxime Allard. Very interesting talk. If acetoglastic methanogenesis is increased by the higher activity through the Lyondel pathway, how does this help increasing the methane fraction in the anaerobic digestion process?
That's a very good question, and one that I'm not necessarily the best to answer. We were primarily focused on the biochemical processes, but I would be happy to get back to you during the later session or by email, and then have a quick chat with one of my collaborators that knows much more about that than I do. I do apologize.
It's good. Maybe we can come back to it during the discussion. Those were the questions now. So I think we can move on and continue the discussion later on. So next we have an online presentation, I think. Willian, I don't know if you're there. I hope so. Willian, are you online?
Yes.
Ah, sorry. I think you are muted. Could that be?
Can you unmute your microphone? Or Zeb from Dryfta, is there a general problem with microphones? So, because when you join the platform, William, you have to allow your computer to share your microphone on top of your video as well. So maybe try to go out of the platform and come again. We are just waiting. In 1 minute, it should be solved, I think.
Yeah, I can maybe already introduce the speaker. Yeah. Yeah, so we have Viliana Vieira Macedo from University of São Paulo. And if we can make things work, she will tell us about the long-term degradation of TBBPA at environmentally relevant conditions and the effect of this compound on microbial community. So let's see if we can get Vilian back in the session. Otherwise, we have the pre-recorded version and we can play that.
I think, William, are you here? We see your acronym. We cannot hear you still.
Maybe needs a minute.
We see your video now. Can you speak? No. Is there a possibility that we can play your pre-recording? You agree? It's less conference experience, but I think it's the only way that we can solve that shortly. And then we try to find back for the discussion afterward. Yeah. Thanks.
Hi everyone, my name is Williani and I'm going to briefly present to you the results that we got from our study entitled Long-Term Degradation of TBBPA at Environmentally Relevant Conditions. the effect on the microbial community. This study is part of my PhD study, and it's a collaboration between the University of São Paulo and Aalborg University. In my PhD, we are addressing the problem related to the presence of organic micropollutants in wastewater treatment plants, especially the pollutant called TBBPA, tetrabromobisphenol A, which is a flame retardant applied to meet fire safety requirements into all sorts of materials, such as plastics, building materials, automobile, electronics devices, and such. And this pollutant has highly immunotoxic and neurotoxic properties, and it's bioaccumulated in the food chain. So in previous studies, we have found out that TBBPA bioconversion in continuous systems in domestic sewage occurs during acidogenesis. But then we wanted to investigate how the microbiome would be affected by long-term exposure. So we had an acidogenic system in which we operated over 100 days in conditions and COD conditions related to the domestic sewage. And then we would sample the biomass weekly, and then we used the amplicon sequence of the bacterial 16S RNA gene to see how the microbiome changed over time. And the highlight of this work is because we have addressed this problem at environmentally relevant concentrations of 50 micrograms per liter, and most of the studies regarding the degradation of micropollutants use unrealistic concentrations, which leads to different results towards the behavior and how the microbial community works. So we could see that the major temporal changes occurred in the first 20 days and was most likely related to the acidogenic condition of the reactor rather than the pollutant itself. But we could see a shift in the bacterial composition from the 6th to the 8th week. And then in this shift, we could see that Enterobacter klebsiella decreased in the relative abundance, and so did the TBBPA removal. We also have seen that the dominant genera in the microbial community were Clostridium, Sporolactobacillus, Enterobacter klebsiella, and Salmonella. And we had the TBBPA removal efficiency higher than 99% from day 59. And then when we correlate the presence of these organisms and the physicochemical parameters that we analyzed in this reactor, we actually have seen that Enterobacter and Clostridium were the only two genera that were strongly and positively correlated to TBBPA biodegradation when we use Pearson's coefficient correlation. And what that means is that even though these results point to a specific stage of the anaerobic process in which TBBPA is bioconverted, and to the possible candidates, the putative candidates responsible for the pollutants breakdown at environmentally relevant concentrations, the enzymatic mechanisms by which this conversion occurs remains a mystery. And that's why we are conducting proteomics and protein seq experiments afterwards. But when we correlate the results, the physicochemical results, with the behavior, with how the microbiome changed over time, we could actually point out to which organisms were involved in the degradation of this compound. And to wrap it up, how relevant is this? How can we use this knowledge? So the relevance of fundamental research on how a micropollutant is biologically converted is the first step to apply biochemical knowledge on industrial processes. And knowledge on TBBPA degraders in environmentally relevant conditions leads to a better understanding not only of how TBBPA affects the ecosystems, but towards other flame retardants. And with that knowledge, we can, in the future, optimize the wastewater treatment systems. I would like to thank you, the two groups that I'm part, both at the University of São Paulo and Aalborg University, and also thank you, the São Paulo Research Foundation, for the funding for this work. Thank you.
There's a Larsen somewhere in the room. Congratulations, Mira. It's funny because Larsen is also a name in Denmark. No? So the degradation of these complex compounds is relatively complex. It's interesting to see how you can address that functionally in the microbiome. Vilian, are you able to speak or not?
I think I have figured out how to unmute my microphone by now. I'm so sorry.
We can hear you. That's perfect now. We can redo your presentation if you want.
I'm happy.
So in the chat, is there somebody in the room who wants to ask a question on this complex component? I have a rapid question. You identified some organism like Klebsiella and others that may be also antibiotic resistant. We will see that probably tomorrow in the sessions on resistance, but does it mean that the organism that can degrade these compounds are the ones that are resistant, or does it mean that an organism that is resistant to a complex component, a xenobiotic substance, is able to degrade it always?
It's really hard to tell, and I would say no, especially because with the results that we have come up with in the further experiments that I've mentioned, which was in the new version of my presentation, but I'm just gonna briefly describe it. So the answer that I would give right now, it's no. One thing doesn't mean the other, especially because we need to go into details to see what's the metabolic route for the degradation, which metabolites are being formed, and which genes are being expressed. So in the protein SIP experiment that I've mentioned, we actually were able to confirm the correlations that I've just presented. So we were able to associate labeled proteins to specific organisms. They are from the same family, as I have mentioned. So they are both from Clostridiaceae family and Tyrobacteriaceae family. And that's the next step. It's because with the correlation, when we correlate the relative abundance of the microorganisms with the removal of the pollutant, we have an idea of which organisms are actively degrading. But with the protein SIP, we can actually point out, because we fed the microbiome with 13C labeled TBBPA, and all carbons that were labeled were the carbons in the aromatic rings. So they must have been necessarily opened so the proteins could be labeled. And then one study confirms the other. But we haven't yet got to the point where I can describe the metabolic route. And therefore, I can't say that because they are antibiotic resistant, they are necessarily able to degrade any micropollutant. I don't know if that answers your question.
Perfectly. Thank you very much. And maybe a question, not to answer now, but toward the discussion to you and Nadi. It would be, how can you translate this protein labeling measurement into rates and yields that can be used for process design and control afterwards? So try to think about that, and we'll discuss that in the roundtable after the last presentation.
Yeah, so lastly, we have Holly Adejumo from University of Michigan. I think it's a pre-recorded.
She will be live.
She will be live, okay. Yeah. Holly, are you there?
Hello, I'm here. Can you hear me?
Yes, perfect.
Okay, great.
So Holly will talk to us about the haloacetronitrile biotransformation in human gut culture. So a little bit different topic to finalize.
Can you share your screen? So if you move your mouse, you should be able to find the button share screen. We can see it very well.
Perfect.
Very good. And we can hear you normally. Nice. The floor is yours. We cannot hear you.
Okay.
Now we can hear you. Yes, this is very good.
Okay, great. So hello, my name is Holly Adejumo. I'm a PhD student at the University of Michigan, and I'm co-advised by Doctors Nancy Love and Laura Rozek. Chlorination is credited with significantly reducing waterborne disease risk and protects people from pathogens. But with every solution, there's a consequence. We see that disinfection byproducts form when chlorine reacts with organics in water. Some studies have suggested that disinfection byproducts are linked to many diseases, such as preterm births and some cancers. There are some substantial gaps in our knowledge around the toxicity of disinfection byproducts. So our goal of this work is actually to investigate the disinfection byproducts that are not regulated, and we hope to close those gaps. The title of the talk is Haloacetonitrile Biotransformation and Presence of Microbial Communities. And in this figure here, you can see we have two sample chromatograms with relative abundance on the y-axis. On the left, we can see 3 different peaks of haloacetonitriles, which are a different class of disinfection byproduct. And background matrices are the components of the sample that we're not looking for. So if we have a background matrix, such as in the figure on the right-hand side, we can see signal suppression of our sample of our analytes of interest. And it can become more difficult for us to detect and quantify the compounds that we're looking for. So let's talk about our process. How did we get here? Our first step was to take a predictive computational toxicology approach. We created this heat map of disinfection byproducts in collaboration with Dr. Anthony Williams at the US EPA. And to orient you, the colors, the red represents very toxic effects, where green is less toxic. And we divided the table into regulated and unregulated disinfection byproducts. And on the top, we have listed different toxic effects. So while toxicity data exists for many regulated disinfection byproducts, we see that less data are available for nitrogenous disinfection byproducts. So overall, through this table, we were able to learn that we have large knowledge gaps associated with toxicity characteristics for nitrogenous disinfection byproducts. So for my work, I'm going to study a class of nitrogenous disinfection byproducts called haloacetonitriles, which are more toxic than regulated disinfection byproducts such as trihalomethanes and haloacetic acids, but they also present a similar occurrence in water distribution systems as the regulated disinfection byproducts. So recent studies have shown that perturbations of the gut microbiome are associated with a wide range of health outcomes. The purpose of this work is to understand the chemical and biological interactions between haloacetonitriles and gut microbiota metabolisms. The first aim, we're looking at biodegradation of haloacetonitriles. So for instance, how are these haloacetonitriles degraded after interacting with the gut microbiome? And what are the toxic effects of the compounds that the haloacetonitriles are degraded into? For instance, if compounds A and B are degraded into C and D, are C and D more or less toxic than the parent compounds? The second aim is looking at how the haloacetonitriles affect functional gene expression in the gut microbiome. So my talk will focus on aim 1, the biodegradation of haloacetonitriles in gut culture. So the methodology for aim 1 involves obtaining fecal samples from humans and inoculating a bioreactor. And then after we establish the gut reactor, we'll monitor for the biotransformation of haloacetonitriles in the reactor. We'll use 2 different instruments, liquid chromatography, tandem mass spectrometry, and gas chromatography mass spectrometry, to both identify and quantify the haloacetonitriles in culture. And we'll also be looking at the biodegradation products in culture. To understand the health impacts, we'll use a computational toxicology approach. So we'll go back to the heat map that I discussed earlier and predict the toxic effects of the biotransformation products and compare it to the parent compound. The methodology for the second aim involves ddPCR and metatranscriptomics. So we'll perform a gene expression analysis and then finally integrative transcriptomics to understand the functional gene expression changes after the haloacetonitriles and the gut microbiome interacts. So like I mentioned, we're going to obtain bioreactor samples and evaluate for the haloacetonitrial concentration. However, we cannot directly inject the samples into the instrument for a couple of reasons. The bioreactor media is aqueous, and the aqueous media is not compatible with our instrument. So a second challenge is also the background matrix, as we discussed in the beginning. The bioreactor media has high levels of compounds such as buffers and other salts, which may create a background matrix. And the microbial culture may also contribute to the matrix effects. So we started to use liquid-liquid extraction as a method, knowing that it can remove matrix effects and concentrate the samples that we're working with. So in liquid-liquid extraction, our compounds start off in the aqueous phase. We add sodium sulfate, which pushes the compounds into the organic phase. And after we extract, we can inject into the instrument. In our case, we're working with 3 different sample types. We're working with water, bioreactor media, and also wastewater treatment plant samples. And we're diluting the activated sludge mixed liquor 100 times. And this is to gain an initial understanding of the microbial matrix effects. The mixed liquor samples act as a surrogate for complex microbial communities. And we assume that the matrix effects will be similar, if not more challenging, to analyze than our fecal microbial community. So we'll primarily be focusing on sample preparation in this talk. And we found that matrix effects were observed when we used trichloroacetonitrile, which is haloacetonitrile, as our model. Here, the y-axis represents the area under each of the chromatogram peaks. And the x-axis represents the concentrations of the compounds of TCAN. So the lower the area count, the harder it is for us to detect the compound. And we see that lower area counts are observed for the bioreactor media. So that's in purple and blue, the 2 lines here. And also the mixed liquor sample in orange, in comparison to the water sample here in green. So this indicates that signal suppression is observed. And this is actually very important for us to understand, because we would underestimate the concentrations dramatically if we don't understand matrix effects. For instance, if we obtained a sample with an unknown haloacetonitrile concentration, and the sample had an area count of 2 × 10⁶, the predicted concentration would be very different for each of the matrices. For instance, the predicted concentration in water would be 100 ppb, whereas in mixed liquor, it would be 650 ppb. So it's important for us to evaluate matrix effects and provide different standard curves for each sample. Our bioreactor is now working. We're working with a reactor called a mini bioreactor array in collaboration with Dr. Vincent Young at the medical school at Michigan. Dr. Young regularly operates these bioreactors. And we selected the mini bioreactor array for a lot of reasons. One is that it's a continuous culture reactor that recapitulates the microbial species that we see in the gastrointestinal tract. It's small volume, so each reactor holds 25 milliliters, and it's high throughput, so we can run 24 samples simultaneously from the same fecal donor and have several replicates from the same run. So we'll obtain the bioreactor and inoculate the reactor with human fecal samples obtained from 15 healthy adults. The bioreactor will be operated at 37 degrees Celsius and pH 7 with an 8-hour retention time. There will be a 2-week bioreactor stabilization period before we add the nitrogenous disinfection byproducts, or the haloacetonitriles, and the reactor will be fed with a defined medium. So here are some unpublished data from our collaborators. On the left here, we have fecal samples with the relative abundance of the genera on the y-axis. We can see that the mini bioreactor array community composition changes with time in the reactor, starting immediately. And after about 1.5 weeks, there's stabilization in the community composition. This change in the community structure informs the operation of the mini bioreactor array for our haloacetonitrile exposure study when we inoculate our samples with fecal culture. Also, we noticed that several major phyla are present, which we would expect in the human gut. In conclusion, we selected compounds that are not currently regulated by the US EPA and that are more toxic than some of the regulated disinfection byproducts. We started analytical method optimization for our compounds of interest, and we characterized the matrix effects of the bioreactor media and mixed liquor. Our next steps for this work are to characterize the haloacetonitrile loss in the bioreactor over time. I want to give special thanks to each of the funding agencies which have supported this work, including the NSF GRFP, and then the NIH F31 Predoctoral Fellowship, as well as the many people who have supported this work. Thank you. Any questions?
Thank you very much, Holly. What I really like with your work is that you bridge the drinking water and disinfection byproducts that will end up in our body. Often we look at the wastewater, the drinking water, the groundwater, but we don't look at the effect in the human body. And your work is one way to go in this direction. You show the complexity of the matrix of the gut microbiome. It's like wastewater, it's also very complex. Like drinking water is complex, everything is complex. You need to rationalize the microbiomes. We discussed that with Takashi before, how to rationalize. But we need also to scale down the experiments. We discussed yesterday that we need to have fast experiments as well, not always to run reactors over 300 days and have profile with a lot of dynamics. We need to see active metabolisms there. And I think with your mini bioreactor approach, it's very interesting, and let's look forward how it develops in the future. Are there some direct questions from the chat? Yes.
Thank you so much for your presentation, Holly. It was very clear. Yeah, I'm just thinking, like, how significant are the levels of these byproducts? How big of a problem do you think this is?
That's a very interesting question. I would say these byproducts are regularly detected in water distribution systems in the US. And in literature, we do see that xenobiotics, so those are compounds that people ingest, do impact the gut microbiome. So this is why we're really interested in answering these questions and investigating the interactions between disinfection byproducts and the gut microbiome. Does that answer your question?
Yeah, sure. There's still a lot to figure out. Maybe small follow-up question. Did you use similar levels in your reactors than the ones that you find in actual water supply?
That's another great question. So what we plan to do is we'll probably start off with higher concentrations than we see in water, just so we can have an easier time measuring the compounds with our instruments and also observing the interactions. And then we'll go down in concentration to what we can see typically in drinking water.
Okay, that's clear. Thanks.
Thank you very much, Nina and Holly. I think we have around 10 minutes, 12 minutes to follow up with a discussion with the participants on all the talks. So if you make a short summary, Takashi helped us to rationalize microbiomes using mags, squeezing the mags and trying to get information on functionalities at genetic level. With Lucas, we see how we can map organisms and their metabolisms in biofilms, like in photogranules as well, and where are the phototrophs located, notably, and what are the interactions with other populations. These interactions were detailed by G about endamo and anamox organisms, and we have seen how we can go one step further with metatranscriptomics. If I understood, with Nadi and William, there was the application of metaproteomics or labeled, so protein SIP, combining metagenomics and metaproteomics to see which population has assimilated the labeled nitrogen or carbon or whatever compound you want to label or atom you want to label. And with Holly, we have made the link between drinking water, the gut, and to look at functionalities more in detail in this complex environment. So we have to close the loop around water across the human environment, across the Anthropocene and anthropogenic metabolisms. And it's very nice to see that we are able soon to make this closure. close the loop. So what kinds of questions we could still ask to the presenters?
I think there's a question that has not been answered yet that maybe is applicable to more than one speaker. It was directed to Dr. Takashi, but maybe other people can answer too. Yeah, this question comes from Dr. Eskiel Santillana. I don't know if you want to ask it yourself, but the question I'm looking at is basically the one, what is your opinion about all the information that is lost when we focus only at the data used to build the MACs? So an example given here is that, for example, genes related to antibiotic resistance or aromatic compounds degradation might be present in the metagenome, but are not necessarily included within a MAC after assembly. So yeah, basically, what's your opinion on that? I don't know, Dr. Takashi, if you want to go first and reply, and then others can jump in.
Can you hear me?
Yeah.
Can you hear me? Okay.
Yes, we hear you clearly.
Yeah. So yes. So this is very important point. I made mistake about possibly. So after the assembly with a very short read metagenome sequencing, so we get contigs at first, and then make mags using binning software. So if you missed some, if you think there are some misinformation, like you may be focused on the antibiotics genes or aromatic degradation genes, so you may use contigs information at first. So actually, I have some experience to find some specific gene, not from mags, but I can find just a contigs. So because the binning process, there are many contigs will be discarded, and then maybe low quality mags are there, obtained or something like that. So you may be careful to check your contigs to find your desired functional gene. Is it okay?
Yes, definitely. Yes. And maybe to come back to this question that I had before, that I mentioned to Lilian and Nadi, what would be your opinion, Takashi? How can we translate all this genetic and metabolic information or functional information into stoichiometric yields and kinetic rates, if we can say like that? Because process design, in the end, it's simply having a rate, having a yield. Yield relates to operational expenditures, rates to capex, so capital expenditures, so the volume of the tank, for instance, and the yield, the operational expenditures, how much biomass is produced per unit of substrate, how much biomass you have to handle in the digester afterward or to treat after the treatment, and how the conversions run in terms of how much oxygen I should put in the system. At the end, if we want to make an impact as systems microbiologists to the engineers, we need to speak the same language. So in a paper, we spoke about a language of love to develop between systems microbiologists and engineers, and I think we need to find common terms. So how can we translate systems microbiology into yields and rates? And other speakers can also answer, and other persons also online. Takeshi, do you have an idea? I see that I should stop my video. Here, there's a problem.
Oh. Yes. Can you hear me again?
Yes.
So yes, this is very critical point, I think. So many engineer side couldn't understand complex microbial ecosystems. So just I want to try simplify the microbiome data to say to make some novel application on the engineering side. So in my experience, so just the EPS or is a common. For example, just the EPS is a common biomaterial in the wastewater treatment process, and the engineer can understand how is a situation, what situation can be, EPS can be increased or decreased or not. So it is connected to the flocculation of the sludge. So I think such kind of understandable information is very valuable for the engineering side. So we may try to translate the complex microbial ecosystem to the relatively easier word for the engineering side. So yeah, that is my opinion. Is it okay?
Yeah, I don't know if everybody agrees. I don't know if there are some people like Michele, Mark, Per. What do you think about this translation into yields and rates? Is it just that these data are there for systems microbiology and we do science to look at the metabolic roots, or can we really integrate this data into yields and rates?
Can I say something? I don't know if you can hear me.
Yes, and then Mark.
Oh, sorry. This is a very interesting topic because it's what we are trying to do with my PhD. We are trying to connect the biokinetics of the degradation of the pollutant that I work based on whatever we got from the protein sip. And the way that we are doing it, it's because from the protein sip, we get some results like the RIA2 value that says how much of the proteins have been actually labeled. And the LR ratio, which says like from that group of proteins, what's the percent that has been labeled. And then if we looked at it from a perspective of a co-metabolic degradation, because I work with environmentally relevant concentrations, so the pollutant is not enough to support the bacterial growth. What we are trying to calculate, combining all of these pieces, is how much of the carbon source we can feed to the system in order to enable the complete degradation of the pollutant. And we've came from like trying to frame which section of the anaerobic digestion actually works on the degradation of the pollutant. So we found out that occurs during acetogenesis. And then we got further seeing that it's during glycolysis. And then further in which groups are working on it. And right now we see which proteins are labeled. And then we are trying just to connect together with the kinetics of the degradation to be able to at least suggest together with the metabolomics of it. So which is the end product? Like how much of carbon source do we need to put in the system in order to enable this complete degradation? But of course, it's super complex because it has a lot of ends into it, and especially because I work with such low concentrations that it's really hard to trace how the degradation occurs. And the fact that we got labeled proteins with such low concentration that we started with is already really nice. So I think that this is related to what you have asked, and I just wanted to share.
Thanks, Mark. Do you agree?
That depends. Yeah, this gives, of course, a lot of information and a lot of, say, research information. But I think the yield and the rates you just get from the traditional process engineering measurements right away. The only thing what could be really useful if there would be good techniques to link, for instance, specific qPCR method on a population to get from the, say, sludge rates to biomass specific rates. And that technology is here and there a little bit present, but that should get more attention, because once you get the biomass specific rates, you can also from engineering point get more insight. Maybe related to that for Nadi, do you think from the measurements you now have been doing that you can estimate the fluxes or how much of the hydrogen added goes directly to methane and how much of the hydrogen added goes through acetic acid to methane? So could you, besides that you see that certain pathways are induced or not, also you have the direct flux analysis, or would you need much more complex measurements for that?
The short answer to that question is that we would need the more complex measurements. The question that earlier made me stumble, how the increase in acetoclastic methanogenesis would contribute to the methane fraction. The reason I kind of stumbled over it is because the answer is very, very complex, and the short answer is I don't know. Because there are, the more substrates we test for this, the more questions we actually get. Like theoretically speaking, you have about 2/3 acetoclastic methanogenesis and 1/3 hydrogenotrophic, I believe. And the theory is that when you add hydrogen, you swap this upside down. And that is actually something you also see in some substrates. When you add hydrogen, you immediately get an increase in biogas production and hydrogenotrophic methanogenesis. But as the substrate adapts, you actually see that acetate is once again being produced. But not every substrate responds that way. So we have no way to generalize what happens when you add hydrogen to a given anaerobic digester. So until we know all the scenarios, we are currently not able to do those calculations yet. I hope that made sense.
Yes, thank you, Nadi. A follow-up of Per. We'll come to you.
Yes, so I would like to add a little bit also to the overall question. It's of course difficult to translate this identity of a MAG and whatever to real stoichiometry and rates and so on. But on the other hand, we are on the way because if we follow some systems for quite some time, we learn about what bacteria are actually present and dominant. And at least full-scale plants in Denmark, we have followed now for 10 years or even longer. It's basically the same bacteria that are present over time. And we now know who they are. We know the MAGs. So we have good ideas about what they do. Then we need to transfer it to rates and stoichiometry. But we can do it by situ measurements. For instance, today it's possible to measure combined fish and microspectroscopy by Raman. That means we can actually measure the pools of, for instance, phosphates, polyphosphate, in different PAOs. And since we know who are the PAOs, we can see we have maybe 4 in this plant, and we can make measurements to say this one takes up so much and this one so much. So we learn about the P release and uptake can be described by those PAOs. So of course, it's not something you can just apply for surveillance, that's all things. But on the other hand, when you know how the different bacteria behave, you may just follow their abundance and learn about it. So I think slowly we are coming. Many filaments, we know exactly what they eat, how fast they grow. So I think because it's not that complex in many plants as we thought, it sounds really complicated when we hear about thousands of different bacteria or 10,000s, but in reality it may be maybe 50 that is important in a certain plant, and we can learn that. And that, as I see it, it's a matter of a few years, and then we can make quite a nice translation of this. And we can now start also making, in situ analysis. I mean, we heard yesterday about nanopore technology that can be done on site, so you can get data from day to day. That means you can actually use that information to try to follow the exact rates and also maybe control it if necessary. So we may hear more about it in the coming days as well.
Thank you very much, Per. And indeed, for PAOs, for instance, there is a saga. So since 1980, people try to think who are the real PAOs of wastewater treatment plants. Maybe with Raman and fish, we can go further with that. And there is still some question between Tetrasphaera and Acinetobacter. So which one is doing the depollution work in the end? Which one really contributes to the dephosphatation? But we are on the way to go. And maybe to close this session, we have here Shaya again, our sponsor from Sweco. And you mentioned that you work on bioremediation. So based on what you have seen here from mostly the wastewater field here, anaerobic digestion and gut microbiota, all these types of analysis, do you think that we can apply that also into groundwater bioremediation, soil bioremediation? How is this very hardcore scientific knowledge being translatable in your daily engineering work?
Thank you for a nice topic, David. Indeed, that's something I've been thinking about the entire conference. And one part which I'm really trying to focus on is less is more. So we need to specify what we are searching for. So I think specifying the goal, what would you like to remediate, in which condition, and for whom, and for which purpose is important before you can select a certain process or scientific findings to add. So there's a reason why we also work from the top-down principle. Because if we go the other way around, we get lost by all the processes which take place. So I think scaling it up via another way, so the top-up approach, is maybe a way we can go by adding stepwise all the fundamental findings that we have. So at first, I think defining the bigger picture, and then like a funnel principle will be more effective for the practice, in my opinion.
Thank you very much, Shaya. And on this top-down, bottom-up approaches that are applicable to microbial communities and engineering and science, I think we should go to it and to refresh. Thank you very much for your presence, Shaya, around this table. Thank you very much, all the speakers. I was amazed by your presentations. Thanks, Nina and Jay, for the interaction. And we see each other again at 12:30 Central European Time, so it means in 55 minutes. And I look at the program to give you some hints, and just to check the timings. Sorry, I have to go to the program to have it through. Do we have it here? Yes. So at 12:30, there is the IWA Fellows meeting. So if there are some IWA Fellows that are present, I think I see two in front of me that are present. So at 12:30, there is a Zoom link for fellows to join and to aggregate together. At 12:30, in parallel, you can visit the roundtable poster discussions. So there it's to collectively speak about the posters between each other. You can visit the sponsor booth of Shaya and Ferrike. And then at 1:15, we'll have the special keynote lecture from Leslie Robertson, who just joined us here, about the Delft historical microbiology. Is it of use for 21st century biotechnologists? And we will see that with Leslie, who reproduced notably the experiments of Anthony van Leeuwenhoek. So have a nice break, and we see each other at 12:30 and 1:15 Central European Time. Bye-bye.